Hello, and welcome to another edition of Biology Explained. Today, we're going to be looking at the fluid mosaic model. Right now, in your body, trillions of cells are making life or death decisions every second. What to let in, what to keep out, and what to fight off. But here's the crazy part there's no brain controlling this. So, how does it happen? It all comes down to something incredibly thin, so thin you can't see with your eyes, yet powerful enough to control everything and anything that enters or leaves your cell. That structure is called the cell membrane. And today, we are breaking down the model that explains how it actually works, the fluid mosaic model. By the end of this video, you will not only understand it, you will be able to explain it better than most students. Let's get into it. Every cell in your body is surrounded by a membrane. Think of it like a border, not just a wall, but a smart checkpoint. It does not just block things, it decides. Oxygen? All right, come in. Carbon dioxide? No, get out. Toxins? Not today. That is how the cell stays alive. But here is where it gets interesting. Scientists once believed this membrane was completely rigid, like a brick wall, and they were wrong. In 1972, two scientists completely changed our understanding of the cell membrane. They proposed something revolutionary that the membrane isn't solid at all, it's fluid. And not just fluid, it's a moving, shifting, living system. Imagine a calm ocean. Now imagine tiny boats floating and drifting across it. That's essentially what your cell membrane looks like at the molecular level. This is what we call the fluid mosaic model. Let's break that down. Fluid means the components can move, they are not locked in place. Mosaic means it's made up of different pieces proteins, lipids, cholesterol, all arranged in an irregular pattern. So instead of a solid wall, think of a constantly moving puzzle. But what exactly is moving? And what are these pieces made of? At the center of everything is something called the phospholipid B layer. Each phospholipid has two parts, a head that loves water and a tail that hates water. So when they're in water, they arrange themselves automatically. Head facing up, tails hiding inside. This creates a double layer, which is a bilayer, that forms the foundation of the membrane. And this simple arrangement, it's the reason your cells don't fall apart. Now, floating within this lipid layer are proteins. These are the real workers. Some act as doors, letting specific substances in and out. Others act as receptors, detecting signals from outside the cell. And here's the key part they are not fixed, they move. Yes, the doors themselves can move. To keep everything balanced, the membrane also contains cholesterol. Think of cholesterol like a regulator. Too fluid, it tightens things up. Too rigid, it loosens things. It keeps the membrane in that perfect middle state. And without that balance, your cells will literally stop functioning properly. Then we have carbohydrates attached to proteins and lipids. This acts like ID tags. They help recognize each other. They help your immune system know what belongs and what doesn't. This is the same system that determines whether a blood transfusion is safe or deadly. So let's bring this together. The cell membrane is not a wall. It is a dynamic, flexible system made of lipids, proteins, cholesterol, and carbohydrates, all moving and interacting. That's the fluid mosaic model. But here's the real question. How does this structure actually work in your body? How does it control nutrients, signals, and even diseases? Let's break that down next. Now, here's where things get interesting. Everything we just explained, it's not just theory. It's happening in your body right now. Every second, your cells are absorbing nutrients, sending signals, and defending against threats. And all of that depends on one thing, the fluid nature of the membrane. So let's break it down with real life examples you'll never forget. Let's start with something simple, oxygen. Right now, oxygen is entering your cells without any effort. No energy, no assistance. It just slips through the membrane. Why? Because the lipid bilayer allows small non-polar molecules to pass through easily. Now compare that to water. Water doesn't just move randomly. It often uses special channels called the aquaporins. These channels act like express tunnels, allowing water to move quickly in and out of the cell. But here's the twist. Not everything gets this easy access. Take glucose, for example. Your body depends on it for energy. But glucose cannot pass through the membrane on its own. So what happens? The cell uses specialized proteins, transporters, to bring it in. And here's where it gets even more fascinating. After you eat, your body releases insulin. Insulin sends a signal to your cells, bring the glucose in. In response, the cell moves glucose transporters to the membrane surface. Once they're there, glucose can finally enter. That movement of proteins, that can only work because the membrane is fluid. Now let's talk about communications. Your cells are constantly receiving messages, hormones, neurotransmitters, chemical signals, and all of that. Your nerve cells use this to release neurotransmitters. Your immune cells use it to engulf bacteria. If the membrane were rigid, none of this would be possible. So now you've seen how the fluid mosaic model explain real life processes. But how did scientists actually prove all of this? Let's look at experiments that changed everything. Before the fluid mosaic model, scientists believed the membrane was rigid, a neat, structured sandwich lipids in the middle, proteins on the outside. Simple, clean, and completely misleading. Because when they tested it, the evidence didn't match. Then came one of the most famous experiments in cell biology. Scientists took two different cells, one human, one mouse. They labeled their membrane proteins with different colors, one green, one red. Then they fused the two cells together into one. At first, something predictable happened. One side was green, the other side was red. But then something unexpected happened. The colors started to mix. Slowly but surely, the green and the red spread across the entire membrane, blending together. This meant one thing, proteins were moving. And if proteins can move, membrane cannot be rigid. This single experiment provided direct evidence that the membrane is fluid, not static, not fixed, but dynamic and constantly changing. Scientists didn't stop there. They wanted to see the membrane. So they used a technique called freeze fracture microscopy. They froze cells, then split the membrane apart. And what they saw was shocking. Instead of smooth layers, they saw tiny bumps scattered everywhere. Those bumps were proteins embedded inside the membrane. This was proof of the mosaic nature. Proteins weren't forming neat layers, they were scattered like pieces in a complex puzzle. But one question still remained, how do these molecules actually move? To answer that, scientists used a technique called photobleaching. They tagged membrane molecules so they glowed. Then they used a laser to erase the glow in one small spot. The spots went dark. Now here's the key moment. If the membrane was rigid, that dark spot would stay dark, but it didn't. Over time, glowing molecules moved back into that area, and the light returned. This showed that molecules We're constantly moving within the membrane. This confirms again that the membrane is fluid. And with that technology, we've discovered something even more interesting. There are special regions in the membrane called lipid rafts. These are organized zones where specific proteins gather to perform tasks. So even with the fluidity, there is structure. It's not just random movement. It's called controlled chaos. So now we've seen the evidence, we've seen the experiments, we've seen how scientists proved that membrane is fluid and mosaic. Here's a few important questions and some common misunderstandings we need to clear up. Let's wrap everything together. At this point, you understand the structure, the function, and even the experiments. But there's one thing that separates average students from top students. They understand the why behind the details. And that's exactly what we're about to clear up right now. First question: is the cell membrane completely fluid? Yes, but not entirely. Molecules can move sideways, but not freely everywhere. Some proteins are anchored, some regions are specialized. So it's fluid but controlled. Think of it like a busy city. Movement everywhere but still organized. Next question: why do we call it a mosaic? Because it's made of different components: lipids, proteins, cholesterol, carbohydrates, all arranged irregularly. That randomness is what gives it flexibility and function. No two areas of the membrane are exactly the same. Another common question: do proteins flip from one side of the membrane to the other? No, proteins are usually fixed in orientation. Their position matters for their function. If they flipped randomly, the cell will lose control. And in biology, positioning is everything. Now, here's the critical one: what happens if the membrane loses its fluidity? If it becomes too rigid, transport slows down, communication breaks down. If it becomes too fluid, the membrane loses stability. So the cell must maintain a perfect balance. And when the cell balance is disrupted, diseases can happen. In conditions like cystic fibrosis, membrane transport proteins don't function properly. In neurological disorders, membrane signaling is affected. Even viruses take advantage of membrane flexibility to enter cells. So this isn't just biology, this is your health. Let's bring everything together quickly. The cell membrane is not rigid, it is fluid. It is not uniform, it is mosaic. It is not passive, it is active, selective, and dynamic. If you remember just one thing from this entire video, remember this: the cell membrane is alive with movement. It controls what enters, it controls what leaves, it allows to communicate, adapt, and survive. And without it, life as we know it would not exist. If this helped you understand the fluid mosaic model better, then you're already ahead of most students. And if you want more deep, simplified biology like this, make sure you stay tuned for the next lesson. Don't forget to follow and subscribe. Like this video.